Hello, folks. Hello and welcome to the Apologetics Academy, which is a weekly interactive online apologetics webinar, which I run. My name is Jonathan McClatchy. I host these webinars, and the idea is that every week we bring on different scholars, scientists, philosophers, thinkers from across the theological and philosophical spectrum to present on topics of interest to Christians, especially those topics that concern whether or not Christianity is true. And we also investigate other worldviews and religions and so on as well, such as Islam, Mormonism, and so forth. And we're also interested in questions of design in nature, which will be the topic of our discussion today. So we normally have a speaker give a presentation, and then we open up to open floor Q&A discussion. There's a number of ways you can participate. You can submit questions in textual form by hitting the Q&A button at the top of your screen. You can submit anonymously if you wish. You can also click the raise hand button at the top of your screen, and at the appropriate time during the Q&A, I'll be happy to promote you to panelists to ask your questions of our guest speaker. You can also participate in the chat as well. So just to introduce tonight's speaker, we are very honored to have with us Dr. Matti Leisola from Finland, who is a biologist, and he is the author of the book Heretic, One Scientist's Journey from Darwin to Design. So without further ado, I'm just going to turn over to Dr. Leisola. Maybe he wants to say a few words about himself, about his background, and just launch into the presentation. That would be awesome.
Okay, thank you for inviting me here. This is a new experience to me. When I was studying, there were no computers at all. I was born already 71 years ago. Coming next week is my birthday. I have worked a long period in universities and in private industry. My basic education is in biotechnology, and I studied chemistry and biochemistry. After my doctoral thesis, I moved to Switzerland, where I worked 7 years at the Swiss Federal Institute of Technology in Zurich area. I was called to come to a private industry, and I worked 9 years as a research director in biotech industry, and had a lot of connections to the United States. And we were, for instance, making a joint venture with Eastman Kodak and put our biotechnology to the same company. And the name was then Glencore International. The name has changed, and Glencore International has been sold to DuPont. And I was a research director from Finnish side and traveled often to the United States. So I have experience in industrial biotechnology, both in universities and in industry. I thought that I was doing very practical application-oriented work in university, but moving to industry, I realized how big the gap was between university and industrial research. I want to share with you a little bit of my book, and I start with a PowerPoint presentation now. I only showed you my face first, that you see how I look like. Now we go to PowerPoint presentation. Here is the book, Heretic. This is the name given by the Americans. I didn't necessarily want to be called a heretic, because I don't think I'm a heretic. I think many more people are heretics than I am. I originally wrote a book 5 years ago in Finnish language, which is now in 4th edition. I translated the book very quickly into English, and then Jonathan Witt helped to put it in proper, nice English and helped to reorganize it a little bit. And I thought that I am quite good in English writing, but with Jonathan Witt, I understood that I'm not. English is not my native language. I started with Finnish. I must congratulate Jonathan that he could almost pronounce my name properly, because usually Americans pronounce it in a very funny way. But in Finnish language, you always have the accent of the first syllable, so I'm Leisola, not Leisola. Heretic, One Scientist's Journey from Darwin to Design. It tells you already that As part of my education, I swallowed the Darwinian story, the naturalistic view of our society and so on. I didn't stop to think about these questions before I was already studying biochemistry in university. I tell you a little bit what I have been doing in science, and I do that with one picture. You can probably see something in the corner, and that's a seed. And out of this seed grows a birch tree. And all this growing is a huge amount of information processing. Birch tree is the natural tree of Finland. I have many birch trees in my garden. And I've studied actually the biodegradation mechanisms of birch tree. This picture may seem a little bit complicated, but if you start here, you can see part of the trunk. And if you take a slice of that, you can see the annual rings. You can see the cells where the water goes up. You can see the phloem cells where the nutrients and sugars come down. And cambium cells, which is a very thin layer where actually the growth occurs. So most of the tree is not growing, it's only this cambium layer that is growing. And when you go to the tree, actual tree, you get tree cells and you get cell walls. And the secondary cell wall is rich of polymers. And there you have cellulose, where you make your paper. There you have xylan, which is another kind of polymer. And then you have lignin, which is a glue material that glues together these fibers. I've been studying the mechanism that fungi use to degrade cellulose, xylan, and lignin. I spent almost 40 years in these studies. Cellulose is made of glucose molecules, xylan is made of xylose molecules, and lignin is the funny compound. I have written a paper that was published in Biocomplexity 2012 about my thoughts about lignin, which is a very curious molecule. And the name of the paper is Lignin Designed Randomness. And there I explain all this in more detail. So my specialty are cellulose, xylan, lignin degradation, biological degradation mechanisms and enzymes related to that. But then I have also worked with sugars and specialty sugars and sugar alcohols. And I started with a Japanese professor about 18 years ago on International Society of Rare Sugars. And we have been looking at different applications and production methods and medical applications of non-natural sugar molecules. So this is very much my background. And if you want to understand more, you can go always to this article, which is, I think, the best one I've ever written. Now to the book. My book Finnish name was here. And if I translate it directly, it means in the wonderland of evolutionary faith. And in the background, you see Alice in Wonderland book cover. You see Darwin looking at me angrily, and I am in the front here. It's now in the 4th printing in Finland. We have sold about 2,500 copies. People keep on buying it, reading it, and commenting it. Finland is a very small, we have a very small population that if we would sold relatively as much books in United States, that would be 150,000 sold copies. Alice in Wonderland, I just looked at Google, it says about Alice in Wonderland that it is considered to be one of the best examples of the literary nonsense. And this is after my long research career and hundreds of discussions with colleagues and students, my view of grand evolutionary theory. What does my book then contain? After the introduction, it has 12 chapters, and I go very quickly through what these chapters are talking about. Suspicions Awoken talks about my study times when I was studying chemistry and biochemistry, reading books about enzymes, and always in those books was the last chapter about origin of life, origin of enzymes. The big textbook was Dixon and Webb Enzymes, and the last chapter there was Origin of Enzymes, and the chapter had several times mentioned that there are huge problems. Nobody understands how enzymes originated. That stopped me first time to think about what's happening here. My study colleague came to me one day and said that this book tells about the same things that you are thinking about. At that time, I met Melvin Calvin, who came to my lab. He was a Nobel Prize-winning chemist who studied the photosynthetic pathway in green plants using radioactively labeled carbon dioxide. One important person I met at that time was Francis Schaeffer, an American philosopher and theologian, came to lecture in Helsinki University. At that time, I had no understanding of philosophy, and his lecture really stopped me. I bought all his books and started reading philosophy that I thought that was very little value at that time. Another person I met was Professor Wilder Smith, originally a British professor who lived in Switzerland, had 3 doctor titles, and somebody gave me his book, Origin and Destiny of Man, and it had a big influence on me. First time I realized that biology is information-based. Later I met in Switzerland another Nobel Prize winner, Dr. Werner Aber, who got the Nobel Prize of his work of genetic engineering tools. And to my amazement, he also considered creation as the most plausible explanation for life. The second chapter is about fossilized materialism. There I talk a little bit about the character of fossil record, but since only 2 weeks ago, Günther Pechli was talking about that, I am not going to talk about that. And Paul Nelson was talking about common descent in July, and his talk was also excellent. I just listened to it yesterday. In this chapter, I talk about influence of paradigms and how to interpret circumstantial evidence like fossil records. Chapter 3, students began listening. I invited students because they were interested to hear my views on evolution to my home in 1976. And in our small apartment was 67 students sitting on tables and chairs and under the table and everywhere packed. And I realized how exciting this topic is. 1978, I was an acting professor of biochemistry, and I discussed about my views of chemical evolution, and students were actively participating in discussions. And then in Switzerland, I was teaching enzyme technology, and again, students were motivated to talk. And I often had some of the students coming to make their master's and doctoral thesis with me. There was a tremendous interest in that. In this chapter, I shortly talk about mutational evidence of Darwinian mechanism. Professors and presidents react. When I started to openly criticize the Darwinian views in Finland, Finland is a small country, everybody knows each other, some professors more or less attacked me directly. But some professors called me privately and said, keep on pressing, asking questions. That's good. It's good that somebody finally asked these questions. My professor of electrical engineering was ready to come to the Finnish television to say that to him, Darwinian story is is standing on clay feet, and it has very little intellectual evidence. I organized a trip for Professor Wilder-Smith in 1981 to Finland. We traveled through 5 Finnish universities, had a television program, press conference, and it caused a huge turmoil in Finland. People were sending letters to the editor of the newspapers. Altogether over 300 and 200 were published. Then I realized how hot this topic is. In 1999, I was organizing a seminar and asking the question, the worldview nature of evolutionary theory. And that shocked the president of Helsinki University, and they almost canceled the seminar and said that if we organize anything like that, we have to have both sides. So I started to plan 2004 a seminar where I invited Paul Nelson from the United States and Richard von Sternberg for the design side, and 2 best-known evolutionary biologists in Finland. And we had planned a 2-day seminar. But then the professor started to react angrily, and I described in my book part of the email discussion within the professors, and finally the president canceled the whole seminar, although the American visitors were already coming to Finland. Again, it showed me how hot the topic is. Publishers hesitate. I translated the book by Wilder-Smith, Natural Sciences Know Nothing of Evolution, and the major publishing house in Finland published the book, but it was attacked so heavily that the publisher got scared. After selling about 700 books, they burned the rest. And the pressure was so strongly against that. Broadcaster bias is the next chapter, and there I explain how a group of young people, probably about the age of Jonathan, who is hosting this whole session, 4 of them wanted to make a film, and they went to Finnish broadcasting company and got some money and promised that they can make the film. The topic was more or less taboo originally. They made a film, 30-minute film, The Deep Waters of Evolution, where I was interviewed, German professor Sigrid Scherer was interviewed, and one professor from the United States. All of us were critical of the mechanism of evolutionary theory. But the broadcasting company hesitated to show the film. They waited for 2 years almost before they showed the film. You can see that now in internet, if you want, with the name of Deep Waters of Evolution. And they showed that on Good Friday, giving the message that this is religious propaganda, although it was directly scientifically based criticism of Darwinian evolution. Church Evolves chapter talks about our Lutheran church. I've met at least 4 of the bishops of the church. Most of them had attacked me and said that I mixed my religious belief with science, and I have tried to convince them that I am purely scientist. I give scientific criticism. They should support me and be happy, but that's what they have not been. Rationalist behaving irrationally is a chapter where I tell about all kinds of attacks I have been facing and some other people have been facing. Finnish Skeptic Society has, for instance, given 2 times a pseudoscience award, not directly to me, but to my laboratory, and then to a publishing company who published Wilder-Smith's book, giving the message that I'm a pseudoscientist. Chapter 9, Colleagues Dare to Explore, talks about my time in Switzerland, my unusual experience with colleagues there. We had discussions during the coffee break practically every week about the Darwinian mechanism with my colleagues who were bioscientists. And many of them were skeptical about Darwinian evolution. My professor there got angry with my first seminar where I explained why I don't accept the Darwinian message, but he became a good friend of mine later. And I met the dean of biological faculty. The dean, Professor Winterhalter, said that Dr. Leisola, you're a very religious man. I said, so are you, Professor Winterhalter. Both of us have worldviews that we can prove with scientific methods, and we base our thinking on our worldviews. And Professor Winterhalter said that you are right after a small pause. The chapter Chasm Widens talks about the problems. It talks about paradigms, worldview effects, phlogiston theory. It wonders why science can be used to support atheism in universities and not theism. In Helsinki University, for instance, we have had Richard Dawkins, we have had Stephen Jay Gould, we have our own local Dawkinses and Goulds who actively promote atheism, and that's no problem. But when I tried to organize seminars to discuss the matters openly, the seminars were cancelled. And the final chapter is then discussing what is the final reality. Has the universe and the life created itself? Is everything self-created, or is there something behind everything else? This is not a new question. It's not a result of American creationists. It's a question that already Greek philosophers were pondering. Socrates, for instance, considered that a supreme intelligence is behind everything. Now, when I'm retired, I've been reading a lot of Greek philosophers this year, and it's interesting how they struggle with these questions. Okay. Chapter 10 is here, written in red, and I will go a little bit deeper on this, if you still bear with me. I ask a question: have scientists already been able to surpass the capabilities of evolution? This may be a strange question. If I and you are products of evolution and we are not able to produce even one living cell, how can I ask a question like that? I would actually say that if all the scientific minds and all the scientific tools and laboratories would be put together, we still couldn't create even one living cell. This title is actually invention of my biochemist colleague Branko Kozulec. He's from Croatia. And we have written a paper that has both science. It criticizes a Nobel Prize winner, Jack Szostak work, who has tried to prove that random sequences can have activities. We submitted this paper first to a biojournal, but it was said that it has too much philosophical aspects. Then we sent it to a philosophy of science journal. They said it has too much science, and they would never publish a paper that is critical to Darwinian evolution anyway. Now it's openly readable to anybody in this address. I consider this one of the most important papers I've been involved with, and I'm really thankful to Dr. Kozulec, whom I met in Switzerland and who helped me to understand some questions more deeply. So I am claiming that we scientists have done much more than evolution can ever do. I start with a sukiyaki party in exactly 30 years ago in November. Around this time I was sitting in Kyoto in a chair that doesn't have any legs. I have knee problems anyway, and after 4 hours sitting here I had real pains. There I met Professor Yamada, who is a world-famous fellow because he developed the microbial method to produce acrylamide. And during this meeting, he asked me, Dr. Leisola, what do you think about protein engineering? That was a new emerging field where you modify proteins and especially catalytic enzymes. And I had to say at that time that I have no experience of this field. I have been working with enzymes, but not with modifying their properties. We were walking in Kyoto in the Golden Temple area, and Professor Yamada said to me that I do not believe that proteins can be improved. I believe in God who created them perfect as they are. And I couldn't comment on that. I only said, let's see what happens. As you see, my hair color was different than it's now. Professor Yamada was a very charming person. Let's look a little bit of enzyme catalyst. I worked in enzyme industry, so this is my field really. Enzyme catalysts are widely used in various applications. In washing powders, you have, for instance, protein degrading enzymes that if you have blood stains or other proteins on your white shirt, these enzymes will degrade them and solubilize the proteins, and you get a clean, nice shirt. You add enzymes to animal feed. They help the animal to digest the food better. You have enzymes in high fructose corn syrup production. In the United States, especially, you start with cornstarch. You produce glucose, and then you turn glucose to fructose via an enzyme. Enzymes are used in dairy applications, in cheese making, and you produce lactose-free milk with enzymes that degrade lactose. They are used in baking, at least in Finland. Rye bread remains soft many more days if you have enzymes there. They are used in chemical industry to produce chemicals. We have a problem because natural enzymes are not always well suited for industrial applications. The problems may be with the stability, temperature, pH, solvent stability, reaction rate is too slow, specificity is not what is expected. Something may inhibit the enzymes, or the enzyme need activators, their pH optimum range is wrong, or they are not enough active in low temperatures. So that's why you want to do protein engineering. How to get a better enzyme is then the question. The first thing that you do is find a better natural enzyme in nature. If you, for instance, want to degrade dextran polymer, which is a problem in sugar factories, you go to sugar factory area, try to find microorganisms that degrade this dextran polymer. I worked in a connection of sugar factory, and we isolated more than 400 bacterial isolates that are able to produce polymers and are able to degrade these polymers. But then you can improve existing enzymes. Chemical methods, immobilization was a technique used, still used in some cases. You can cross-link enzymes together. You can crystallize enzymes. My team was one of the few ones that was studying protein crystallization cross-linking as a stabilization method. And we developed some new techniques to crystallize protein, which is not an easy task often. But the modern way is to use genetic engineering methods. You can do specific genetic changes, or you can do random mutations. I take an example. No, I don't take an example yet. First, we have a protein engineering problem here. We do not know how amino acid sequence affects enzyme performance. Enzymes are too large molecules, and it's too difficult so far to more or less forecast what kind of performance you get with a certain kind of sequence. And the protein sequence space, the possibilities of all protein structures is unbelievably big. We have 20 amino acids, and average enzyme is roughly 300 amino acids. So you can see that the possibilities are absolutely beyond understanding and imagination. We don't know how to find the design sequences in random. So that's why you never start building an enzyme or the gene coding it from nothing. You start with existing enzymes. I use an example here, xylanase enzymes. You probably don't remember anymore, but birch tree has xylan fiber that is degraded by xylanase enzyme. Birch tree has about 25% of this material of dry weight. And this xylanase enzyme, I started to work with that. Here on the right-hand corner, you see a fungus that produces this enzyme. It's a wood-rotting fungus. I produced this enzyme already in 1974, and later then started to look at applications. I sent one of my students to University of Vienna about almost 30 years ago, and she isolated the gene for this. So we sent the gene to our American colleagues, and they transferred this gene to a production organism to overproduce this enzyme in large quantities. Then my student went to another university, crystallized this protein, and determined its three-dimensional structure. And this is roughly how it looks like. It's like an open hand. And the active center here, two acidic amino acids, and here xylan fiber. You can imagine that xylan fiber is drawn into this active center, and then it's split into individual X sugars, meaning xylose sugars. This enzyme is used in animal feed, in dry bread, in fruit juice manufacturing, and especially nowadays in biofuel production, bioethanol production, which is a hot topic in this field. This enzyme has some weak points. One weak point is this helical structure here. Another one is in this terminal end. And when the temperature rises, the enzyme starts to open like a ball of yarn. And when the temperature goes back down, the enzyme does not get to the original position. It starts to give wrong structures and aggregate and precipitate. So it's a problem when animal feed manufacturing use high temperatures, something like 80, 90 degrees, and this enzyme starts to die at 50 degrees. But we knew what the weak points are, and what we did is, this is another angle of the same enzyme. We built in these red structures, so-called disulfide bridges, to stabilize the protein. And the temperature stability increased up to 25 degrees with this stabilization. But the activity still remained the same, since we didn't touch the active site canyon at all. One of our mutants with only one disulfide bridge was patented and has been used about 20 years already by an industrial enzyme company, and they said that that was the best modification they had. That is another possibility. What I showed you is actually targeted mutagenesis of specific points once you know the enzyme structure. This year, Nobel Prize was given to 3 persons, and one is Frances Arnold, of his work on directed enzyme evolution. And Arnold herself, and especially the press release, says that they are using the Darwinian method, the natural innovation system, to create new enzymes. And here I totally disagree with that view. I've written a couple of articles about that already in Finland, and actually one was published by the CNS News also. The word directed and evolution are contradictory. It's an oxymoron to say directed evolution. Directed means direction, design towards a purpose. Evolution, by definition, is random, going nowhere. I shortly explain what this means. It's not starting from scratch, creating enzymes by Darwinian method. The starting point is a functioning gene and protein, and the protein already has a protein-protein interactive capability within the cells. You use tools of genetic engineering all the way when you modify proteins this way. You use, for instance, error-prone enzymes. When the gene is copied, random errors are introduced into the genes. Once you create this gene library, you put them in bacteria, you express your protein, and you choose towards your target. You use huge mutation rates, 10,000 to 1 million times higher than natural mutation rates are. You select towards a goal. You fix all the desired mutants. In nature, even 99% of mutants do not fix into the population. And after several iteration rounds, you get a protein variant which is better suited than the original. What is good in this technology is that you don't need to have the crystal structure of the protein. You just need the key gene and you mutate it, you produce random proteins, and you choose variants from the huge set of proteins. You create thousands and millions of mutants. If you look at the code word xylanase and directed evolution, you can find a lot of papers. I only picked 5 of them. They all said that with directed evolution, you can create an alkali-tolerant thermophilic enzyme, or you can make a thermostable xylanase, or thermostability improvement. You see that the same xylanase structure remains, and you basically, by random changes, stabilize it using huge mutation rate and selecting what you want. This does not mean that Francis Arnold has not done magnificent work with his team and created a lot of interesting molecules. It only means that it does not imitate what happens in nature. So what about Yamada's question? Yamada said that I don't believe that proteins can be improved. I believe in God who created them perfect as they are. After 30 years now, I can say that Yamada was both right and wrong. We can improve proteins for our purposes. But Yamada was right, the proteins in their natural context in living cells are optimal. There are papers that explain that if you mutate enzymes and place them in the natural situation in a living cell, they usually don't function. They create problems. The mutations that are good for industrial applications, practical applications, are not necessarily good in the natural system. This is a good moment to take some water. Time here is already approaching 11 o'clock in the evening. I have had a long walk with my dog. I was sitting in sauna just 2 hours ago. If my face is still red, that's because of the temperature of 80 degrees Celsius for half an hour. I move on. This is the first part of my talk. I hope you still can bear with me. And I go to microorganisms because that's another area where I have worked many, many years with my team, both in industry, in universities. You all know that microorganisms are widely used in biotech and food industry. Here we have red-colored E. coli bacteria, yellow-colored yeast, budding, and here we have wood-rotting fungi, or maybe it's a penicillin fungi on a nutrient agar plate. long before we knew anything about microorganisms, their abilities were used in many different ways. They were used in cheese making, they were used in bread making, beer was made with and all kinds of sour milk products and sour bread applications. One of the most interesting experiences I had in Japan, I visited a very old soya sauce factory. We were walking here. I was really afraid of not falling into these black reactor boxes. How do you do soya sauce? You take soya beans, mix with an aspergillus fungi, and aspergillus fungi makes this messy mixture, which is then transferred to these black jars where you have yeast, and the fermentation starts and takes, in these traditional hundreds of years old reactors, up to 2 years. And after 2 years, you press the black, almost tar-like material, and you get the soya sauce. Of course, the modern soya sauce manufacturing techniques are different, but this is the traditional one. I visited one of those in Japan. This is the xylanase producing fungus that I mentioned earlier. It grows like this on plant material, and microscopic picture is here on the left. Trichoderma fungus is a wood-rotting fungus, and it has an interesting history. During the Second World War, the American troops in Far East were wondering why their tents and their ropes are deteriorating. They were made of cellulose, and this fungus eats cellulose. So Americans isolated this fungus and started to study it in their US Army laboratories. And we found out that this is a fungi, or they found out that this fungi can secrete outside the cell high amounts of enzyme proteins. I worked with this during my doctoral work with these organisms, and I've seen in industrial large-scale production up to 100 grams per liter pure enzyme proteins in the liquid outside the cell, which is close to world record. Traditional biotechnology used living cells without really knowing what is inside happening. Nutrients, E, air, pH, right temperature, and you got products, but very little knowledge. Michael B., he used the word, the living cell was a black box. You put something in, you get products out, but didn't know what's happening. But then we learned to isolate microorganisms and cultivate them in reactors where you have pure cultures. Here is an example of a laboratory bioreactor or fermenter, if you like, where everything is carefully controlled. Agitation speed, temperatures, light, pH, and whatever, amount of nutrients and rate of nutrient feeding and so on. This is typical that my student used when they were studying processes that are used to produce bioproducts. Large-scale industrial production started already 100 years ago, especially during the First World War. Acetyl butanol production was important. They were fuels at that time. In this picture, you see a 350 cubic meter reactor. It's almost 20 meters high, and it is used in fungal production, for instance, of this xylanase enzyme. The largest reactors I know about are about 1,000 cubic meters, which means 1 million liter reactors. And for Americans, if you are listening, about 250 gallons, 250,000 gallons. In this type of reactors, different acids are produced, like citric acid or glutamic acid, amino acids, antibiotic, enzymes, vitamins, therapeutic proteins, but probably not in this large scale. I mean, proteins like insulin, for instance. How to improve the productivity? That's, of course, the economical question. If you want to compete in this industry, you have to produce as much and as quickly as possible. So you again go to find better organisms from nature. You optimize the process conditions. This can have a huge influence on the final production rates. But then you mutate organisms. The traditional method was to use radiation or toxic chemicals, ultraviolet radiation, various toxic chemicals cause different kinds of mutations. And this way you could, for instance, increase the production rates of citric acids, or you could get 1,000, even 10,000 times more penicillin than the natural original organism could produce. This was just randomly mutating the organism. So why does this kind of random method increase the productivity? Do we get more information into the cell? No, we certainly don't. Randomness destroys. And very often it happens so that the cell regulation is destroyed. If you feed into your reactor compound A and you want to produce compound C, typically the compound C starts to inhibit this first reaction. So you never overproduce. Cells don't produce what they don't want. Random mutation breaks this inhibition mechanism, and then you start to overproduce compound C. I've used this myself a couple of times to overproduce compounds, and this is still very much used. But since 1953, we have started to understand more and more about the information processes of the cell. Here, you don't have a sausage. It looks like a sausage. That's what people usually think. It's an E. coli bacteria. Its cell wall has been made permeable so that the whole DNA inside the cell is spread here in the background. This is how DNA looks like in the electron micrograph. I don't want to go to DNA information any more deeply, but we know that it's a coded information where much of the information needed to build up and multiply E. coli cells is. This is one of my favorite simplifications to show how dense the information content is. We know that human DNA weighs about this much, 10 to minus 12 times 6 grams. And if we assume, which is totally a guesswork, that there are 10 million species with the same amount of DNA as we have, we of course know that there are organisms that have much more, organisms that have much less. But this is an assumption. Then the whole genetic code book for all the living organisms would weigh only 0.06 milligrams. And to compare that with the size of the mosquito, which we have a lot in Finland, mosquito is 2 milligrams. So the whole genetic code book needed to go for everything in biological world would weigh only 1/30th part of the weight of the mosquito. I checked with this calculation with Professor Lennox, who is a professor of mathematics, when he visited Finland, and he said yes. So genetic information is extremely tightly packed, and it's much more complicated than we ever realized. Genetic engineering allows us to localize genes, to write genetic language, to multiply genes rapidly, to introduce new genes into a cell. And one of the first experiences I had was an isomerase enzyme which converts glucose to fructose. we had a bacteria that about 100 extra copies of isomerase gene. And during the fermentation process, cultivation process, the organism, when it was multiplying, it was losing this extra information, so that in the end of the process, the cells had practically very little of those genes left. We can influence cell regulation, and we can even construct new metabolic pathways into a cell. This is metabolic pathway engineering. Quickly to this. Cell is no more a black box. Now we know what's happening inside the cell much better than we used. We talk about metabolome, which means all the metabolic compounds inside the cell in a certain moment. We talk about protein, all the expressed enzymes inside the cells, and we talk about genome, of course, the genetic information system. And now we can influence these or look what happens when we do things, changes in the environment, and maybe optimize the production better. Escherichia coli is known to everybody by name. Our intestines have billions of these cells. It's one of the best understood industrial organisms, and it is a basic tool of genetic engineering studies. If you find, for instance, a xylanase gene in your rot fungi, you usually isolate the gene and you put it in E. coli and produce your desired product there. And this is an electron micrograph where the black spots here shown, but the arrows are so-called inclusion bodies full of foreign proteins. E. coli has been bioengineered to produce all kinds of compounds. One of the exciting things I have followed when I worked in the company was indigo blue production by E. coli. The key was an enzyme, naphthalene 1,2-dioxidase. This is the enzyme. It's a huge complex molecule. It converts indo to indigo in the presence of oxygen. You start the cultivation of the cells, and slowly the culture becomes blue. It's a good demonstration for students what can be done. Vitamin C production, fatty acid production for biodiesel, 1,3-propane-diol molecule picture here for polymer synthesis, isoprene production for rubber, artificial rubber production, insulin, erythropoietin, EPO, which is used in certain blood diseases, and unfortunately also some people in sports and skiing have been using this molecule to improve their hemoglobin concentration and oxygen uptake rate. So we have done a lot to bioengineer E. coli. Sometimes you can put, for instance, in isoprene was the molecule that is used for artificial rubber production. You can put whole pathways into E. coli. Here is an example where you take upper pathway from Saccharomyces yeast, or you can take the same pathway from Enterococcus bacteria, and then lower pathway from Saccharomyces again, and engineer E. coli to produce that. My team has worked with xylitol. Xylitol is an artificial sweetener. Production and application started in Finland, and it has been shown that in some cases xylitol can even help to fix small cavities in your feet. So dentists in Finland recommend regular use of xylitol. My team has studied how to produce xylitol in yeasts and in bacteria. We have been engineering the metabolic pathways to produce xylitol. The difficult task was we tried to produce xylitol from glucose. This shows some of the central metabolic pathways of sugar metabolism in the yeast. And we worked a couple of years to optimize xylitol production from glucose. Naturally, we get ethanol and glycerol when the conditions are non-aerobic. When air is there, you don't get ethanol, you get basically H2O2, means water and yeast cells. We realized that our bottlenecks, transport of sugars into the cell, redox imbalance, oxidation reduction imbalance, inefficient cycles, regulatory problems, and we never could get economic ways. We could get something like 2% of the glucose as xylitol, but it was far from what we wanted. So experiences. Metabolism is extremely complex in bacterial level already. There are no such things as simple organisms. Everything is complex or extremely complex. All reactions are interlinked, and a typical cell resists changes. If you modify a cell, your production organism has to be protected. In natural environments, it would lose in the competition because it's no more what it is. It has been changed to directions that is interesting to us and for our applications, but it's not necessarily good for the organism itself. Systematic change of organisms is very difficult. Usually only modest results have been obtained, like indigo, vitamin C, amino acid production, or you increase the quantity of a certain gene so you get more production out of that. Limits of metabolic engineering. If the basic metabolism of an organism is not effective, new production pathways can be constructed, but with considerable intelligent input, and such organisms are usually not viable in nature. I asked the question again, have scientists already been able to surpass the capabilities of evolution? And I would say, yes, we have. We have with proteins, we have with metabolic engineering, and we have found limits to these changes. Bacteria have been used as model system for evolution. Of course, bacteria is the ideal one. They grow rapidly. One generation with thermostable bacteria may take only 10 minutes. And in ideal conditions, you can have huge amount of generations. In 1965, Professor Mortlock, whom I know from my Japan connections, said that Arabacter can learn to use novel sugars. But then when it was studied more carefully, it was again the disruption of the regulatory mechanism. Nothing novel was done. 1976, Professor Hall studied Escherichia coli, and he has published on evolution of a novel beta-galactosidase gene. That means an enzyme that degrades lactose. After careful study, he realized that the Escherichia coli has an enzyme that with one mutation can start using lactose as a food. One mutation in an existing gene is well within the reach of a chance mutation when you have billions of bacteria. 2008, Professor Lenski, who has already done 70,000 generations of E. coli, said that evolution was first time caught in action. When you look more carefully, and I respect the perseverance of Professor Lenski of working so many generations of bacteria. But when you look carefully what has happened, it's usually breaking of genes, losing of genes, and so on, not creation of new genetic information. 2012 Salmonella enterica, first time demonstration of a new gene. And when you read the article, you realize that you have a gene which has two activities, a minor side activity. And what happened, after several thousand generations, these two activities were separated, but they were already existing activities, nothing novel. For microbes, devolution is evolution. This is the article title. Species are in a race to the bottom, deleting the genes for as many costly functions as they can get away with. 2014 Bioscience. And this is what happens. They lose activities in a certain niche environment. And if you take an organism that has lost activities and genes and take it back, it can't recover them. There is no mechanism. Transmutation can't recover the lost activities. This is from Professor Dan Tawfik from Weizmann Institute in Israel, who is looking at the level, the protein proper. He says that nothing evolves unless it already exists. This was said about protein, but it's also true for microorganisms. This is the paradox of evolution. You can evolve when you have something. When you don't have, you can't create it by random methods. If you have a protein with a minor side activity, you can improve that activity. But if you don't have it, random methods can't create it. C.S. Lewis, all in Great Britain know him, said many, many years ago this sentence. What inclines me to think you may be right in regarding evolution as a central and radical lie in the whole web of falsehood that now governs our lives is not so much your arguments against it as the fanatical and twisted attitudes of its defenders. For the last almost 40 years, I have personally experienced these fanatical attitudes. I've been attacked severely, and this is what my book tells about, giving some scientific background. Now I've used more time than I thought that I will use. Thank you for listening.
Excellent, thank you. That was really awesome, really fascinating, and very informative. Folks, if this is the interactive portion of the program, if you'd like to interact with Dr. Leisola and ask any questions, you can submit them using the Q&A button at the top of your screen. You can also click the raise hand button if you'd like to interact and dialogue that way as well. Do you want to maybe tell us a thing or two about your work on lignin, which you published in Biocomplexity, because that was a really fascinating study and has, I think, tremendous design implications. Do you want to maybe say a few words on that?
The lignin is an interesting molecule because everything in nature is usually regular. You can take cellulose, you can take starch, you can take whatever, like proteins or DNA, and you have enzymes that degrade it because they are systematic, regular structures. Lignin is irregular. It's created by enzymes that don't directly make the polymer as all other polymer making enzymes do. It's created by chemical oxidation, by radical reactions. So once you have aromatic molecules, they polymerize automatically in irregular fashion. That's why you don't have a system to degrade it. Lignin is the support material. Animals can't use it. If you have a termite eating your house, termites will excrete the lignin fraction and build their nice homes out of that. No animal can degrade lignin. Lignin has a huge energy content. And the question is, If Darwinian evolution has created everything complex in the nature, starting from flowers to plants to fish to birds, why can't any organism degrade lignin? This is what I have been pondering. You have certain fungi that do degrade lignin, but they need energy to do that. So it seems to be simple. There is the most energy-rich compound in polymer world, lignin, and no organism can use that energy as an energy source or growth source. Lignin is the problem. It has been studied for 100 years to find applications for lignin, and even now a Finnish big paper and pulp company is telling that they start to prepare lignin as a possible material for plastics. But what it means, if we don't have lignin, we wouldn't have humus, we wouldn't have anything. Everything would be eaten up. We wouldn't have trees growing, because if organisms could eat lignin, they could easily degrade the rest of the tree. So lignin is more or less a material that is a total mystery. Why nothing can degrade easily? Why it is created with random method? Why the same enzymes that make it, same type of enzymes degrade it, and how they do that? It's not really understood. I studied 10 years of that problem. Now I lost your voice.
Yeah, sorry, I was muted. Eric Anderson in the chat asks, Matti, when you say there was a gap between academia and industry research, are you saying that industry was farther along or academic research?
I would say that I was in an Institute of Biotechnology in Switzerland, and most of the work was aiming at industrial applications. But when I moved to industry, I realized that universities usually don't understand some of the key things. That means economic aspects and productivity and prices and many of those things. So people are excited when they, for instance, produce indigo blue in E. coli and think that they become millionaires. But when you looked the process economics, you are far away, no chance. We produced xylitol producing mutant, but it was so far away from the traditional method, it was not applicable and so on. So I think those people who have both industrial experience and university experience are rare but valuable. I'm one of the few ones in Finland who has done both careers. That's why, for instance, when I came from industry, I immediately told my team that if you can stabilize xylanase, you can make money out of that. And two of my students have made a lot of money with xylanase mutant.
We don't have any other questions submitted, but maybe we can wrap up here because I know it's getting also late for you in Finland and you probably want to go to bed pretty soon.
So, I hope you understood my Finnish accent.
Yeah. Yes, I really enjoyed it. It's very informative, and I'll put it on YouTube so that many more people can watch it and learn from your knowledge.
Yeah. Okay, I hope you read the book. I don't get really money out of that, but it should encourage people to seek the truth.
Yes, absolutely.
Going after the ruling paradigms and so on.
Mm-hmm. Indeed.
Okay.
All right. Well, have a good night.
Thank you very much.
Right, bye.